What up everyone, it's your boy, Mr. Jennings, here today with another big orange biology. We're doing things a little bit different. We're going to try going back on camera for a little bit. And as you can see on the screen, up here, we got the actual notes handout. Hey, we're trying something new, just mixing it up a little bit and see how this works. Alright, now let's get class started. So class is now in session. And today we're going to talk about meiosis, specifically the first part of meiosis here, which was all about what is sexual reproduction and the actual act of meiosis. So in large part, this should be a review, a recap from your previous teacher's lessons on meiosis. Truth be told, you probably forgot a lot of that. Just how it is. Meiosis is something that doesn't come up casually in conversations. Understandable. Let's actually understand though what meiosis is about and its overall function it serves, and ultimately some interesting things about it. Because what the College Board wants you to understand is how meiosis creates genetic variation. Now, we discussed a little bit about genetic variation already in class, but today we're going to talk about how meiosis actually works and the actual processes that create variation. If you want to look anything in this unit here, it's all about how genetic variation is a result of sexual reproduction. All right, let's get in going. So that brings up the first thing here. What is... See that the camera went crazy. What is sexual reproduction? Now, what is sexual reproduction? Sexual reproduction is a very common process. It's any process that involves fusion of gametes, and we call that process a lot of times fertilization. Now, fertilization is anytime you fuse sperm and egg cells together and you create what's called a zygote in humans. But you can create a lot of different organisms. Now, one thing interesting about sexual reproduction, while it is universally that basic process, sexual reproduction tends to follow into this weird category of being very different. So sexual reproduction tends to vary by species. Some great examples. We've already discussed one in a previous lesson. We've talked about fungi. Specifically the yeast that breed the schmooze, if you will, alpha and A yeast. They're normally in haploid form, and they only fertilize to produce a zygote for just a brief second, and then they immediately undergo meiosis to get back into the normal life cycle. But generally, the overall process, despite being varied by species, tend to follow this general rule. You get a haploid cell, you combine it with another haploid cell, and you're going to get... Now, we may have mentioned this before. When we see that word haploid, what that means is half the number of chromosomes. That's usually represented by all N, one N. And diploid is because it means normal set of chromosomes, or two sets, really. And that's normal for most organisms, and that is 2N. We'll see more with haploid and diploid. Alright, so now here we have the overall process of meiosis is defined as reductive, we're going to put a little parentheses around this, cell division. Should also note, because it is dealing with nuclei, it needs to be noted it is a eukaryotic thing. It's a eukaryotic overall process. Now, meiosis, as you can see on the handout, every step's clearly defined to you. That's part of the reason I gave this handout. Knowledge of individual steps of meiosis really is not what College Board is about. You don't see those kind of questions on the test. They used to be on the test when I was a student, but that was a long time ago. It's been over 10 years. Nowadays, they're more focused on understanding the big picture and the big processes. We'll get more into that as the unit progresses. But we need to understand the basic fundamentals. That's why we're doing this little recap video. So meiosis really breaks down into two steps. And the main thing you need to know is right here at this point, and I'm going to try to draw as straight a line as possibly can on this digital tablet, you're going to get a situation of basically no DNA replication. So after you undergo meiosis 1, as you go through all the ones with the number 1 denotion, you'll undergo not another interphase. You're going to go straight into prophase 2. You're going to skip DNA replication altogether. Hence why you go reductive. Well, you go in, if you want to look at this, typical cells typically go 2N, two in, and once you get to this point right here in telekinesis, you go from half the full set of chromosomes to half a chromosome as soon as you hit telophase 1, and that continues on throughout all the other processes. So you go diploid, 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 all of a sudden haploid. Sorry, this cold will not leave me. But overall, meiosis is a universal process, and you carry out organisms. It has some variations here and there, but it is something worth noting about it is it serves two general purposes. So this is the overall main. Now, meiosis has two main functions. First and foremost, it produces haploid cells. That is its main thing. Fun fact, we believe meiosis actually evolved from mitosis. It's very similar processes, it uses very similar infrastructure, and we know mitosis came first because asexual reproduction is very common, especially the most common form on the planet. By looking at the number of individuals, we know that has to happen. Well, we believe at some point, and we as in scientists, we believe that at some point, we have evidence to support this a little bit too, that when sexual reproduction came around, you needed to make sure you had half the DNA. So when you combine two cells, so you take, a sperm cell and an egg cell, and you combine them, you have the exact same number of chromosomes every time. And in order to do that, you need to have haploid cells. And in order to make sure the cells are the correct combinations, because remember, in humans, for instance, we have 46 chromosomes, but 23 different pairs. So make sure at least one of those 23 pairs, so one representative from each of those pairs, are present in the haploid cell. You need some kind of controlled, orderly process, and that's what meiosis does. It gives you a controlled, orderly process of taking taking diploid cells of those 23 different pairs of chromosomes and making sure each haploid cell, each sperm and egg, gets one of each pair of those chromosomes. And that's a very important process. Make sure everything is orderly and functional. So the main function is to produce haploid cells, but also in the mere process of making haploid cells, it does a secondary function. And that is, it creates unique gene combinations each gamete. So each sperm and egg cell is going to have a unique combination, specifically 2 to the 23rd power. That's a very large number. To give you that, because we've already talked about that in class. That's just for one gamete. And obviously we discussed the other day in class, when you take one individual 2 to the 23rd power and you mate with another individual who has 2 to the 23rd power options, you're going to get a near infinite combination of offspring. And it's pretty interesting. Now let's talk about meiosis in specifics. So this was originally designed to be a little bit more of the traditional PowerPoint notes. So that's why it's phrased this way. Try something different here. So there's a couple key phases that we do need to talk about when it comes to meiosis. First off is Prophase 1. Now Prophase 1 had a very key role of basically this is where crossing over occurs. If you recall, in class we've already discussed this, but crossing over is basically when you have two chromosomes that are homologous with each other. During Prophase 1, when they line up next to each other, they will sometimes get tangled and form what's called a chiasma. Now, when they split apart, you're going to get two new unique combinations. And I should use a different color. Let's see if I can pull down that little menu here that gets annoying. Let's make that one red so it kind of sticks out a little bit better. So we end up getting a whole new combination of genes just by crossing over the chromosomes. And this is a very common process, common enough process. We're going to talk about how do you know when this occurs and what genes it typically affects the most, which are linked genes, later in the unit. When we get to non-Mendelian genetics, towards the end of the unit, we'll talk about crossing over and actually knowing when crossing over occurs and the likelihood of crossing over. It all comes down to location, like with most mutations. But Prophase 1 has that. That's a key note you need to be aware of, is Prophase 1 is when this occurs. You may not see a very specific AP exam question related to that, but it might be something out there on a quiz or something like that. It's a very easy question to ask. Now let's wrap it up here and talk about Metaphase 1. Now Metaphase 1, and let's go ahead and put its sequential phase, Anaphase 1. So Metaphase and Anaphase 1 play a very key role in the process of lining up. So in mitosis, because meiosis, again, very much built on the infrastructure of mitosis, my metaphase, everything lines up single file. And meiosis, homologous pairs, homologous chromosomes line up in pairs. So when they line up in pairs, excuse me, when they line up in pairs, it's drastically different from metaphase and mitosis because everything lines up in single file. So we're lined up 2 by 2 by 2, go on down for 23 chromosomes in a human. Now, converse that to anaphase. So that's what happens in metaphase. I should make that note. This is strictly a metaphase thing. And those keeping notes at home are freaking out. Sorry about that. Now, in Anaphase 1, it should be noted, this is when chromosomes split. Let me make sure it's homologous. The homologous chromosomes split. Much like Anaphase normally does, the chromosomes move away. This one's homologous chromosome splits. But what's notable here is a failure to split. So, there's sometimes a situation where a failure to split occurs. And I should get to there. That's when the chromosomes don't split properly, and this creates a process known as non-disjunction is how you result in disorders and abnormal such as trisomy 21, trisomy 15, Klinefelter's syndrome, and even situations like Turner syndrome. Anytime you have a chromosomal account difference, it's often because of non-disjunctions. And there's a variety of them out there, and we will elaborate more on them as the course progresses. When we especially talk about human genetic situation, and they're very interesting to go into. Alright, well that concludes today's little video. There's going to be some editing on this, so it says like right now it's like 14 minutes, but I got a feeling it's going to be. I hope you enjoy this. This is just a different change of pace. Instead of me doing a PowerPoint, I get on camera a little bit. You can see the Christmas tree behind me. It's the holiday season. Overall though, I hope you like this method. We'll try this for this unit, see how it works out. I'm always ever changing and trying out new things all the time. Alright, well, thank you for watching. Hope you have a great day.